First, one of the most important properties of the plasma membrane is that it exhibits what's called selective permeability. If you think about it, if a college or a club is very selective, they are very picky about who they let in or out of that college or that club. And so the plasma membrane works in a very similar way. The plasma membrane is very picky about what it lets into or out of the cell. So that's one of the most important things to associate with the plasma membrane is that it's selectively permeable. Now let's talk about what is the plasma membrane made of. So the plasma membrane is mostly made up of phospholipids. We need to know that phospholipids, they are supposed to have polar phosphate heads that are hydrophilic and love water, and then they have nonpolar hydrophobic fatty acid tails that can't stand water, that don't like water. And so if we were to dump a whole bunch of phospholipids into water, what would happen is that the nonpolar hydrophobic fatty acid tails would all turn towards each other to get away from the water. And then the polar phosphate heads would turn towards the outside and turn towards the water because they like it. It's very similar to if we were imagining that the phospholipids could talk, it's like the fatty acid tails turn to each other and they say, I don't want to be near the water. Do you want to be near the water? No, I don't want to be near the water either. Okay, then why don't we just turn towards each other so that way neither one of us has to look at the water, touch the water, face the water. Okay, sounds good. So they turn towards each other to try to get away from the water because they don't like it. So that's the first thing that the cell membrane is made of, is phospholipids. We need to know that the fatty acid tails on the phospholipids, they can either be made of saturated fats, saturated fatty acids, or unsaturated fatty acids. If the phospholipids are made up of saturated fatty acids, we need to know that saturated fatty acids tend to be very straight. And because they're very straight, they're going to be able to stack very nice and neatly and be able to form a solid. Saturated fats will therefore help something become a solid very easily. So cell membranes that have phospholipids that are made up of saturated fatty acids will tend to be very stiff and rigid because they form solids very easily. Then cell membranes that are made up of unsaturated fatty acid tails will tend to have a bent shape to them, and that will actually prevent them from becoming a solid. It will prevent them from becoming a solid. And so this means that the phospholipids that are made up of the unsaturated fatty acids will tend to remain more fluid because those phospholipids can't really get that close to each other. They remain fluid. Now, the second thing that the cell membrane is made of is cholesterol. Cholesterol also helps with the fluidity of the cell membrane. So as the cell membrane starts to become cold, what will happen is that the phospholipids are going to want to get really close to each other in order to form a solid. And the cholesterol, almost in a way, is going to be like pushing them apart and shoving them away from each other so that they can't form a solid. So in other words, it's going to help the cell membrane remain fluid. So cholesterol can actually help the cell membrane stay fluid. Also, if the cell membrane starts to become too warm, the phospholipids may melt, but the cholesterol won't. And so the cholesterol, in a way, will help act as support beams a little bit and will kind of help the cell membrane keep its shape without it becoming too soft and too fluid. So it also helps with the rigidity of the cell membrane as well. So although cholesterol is usually associated with something bad, it actually does have an important function in our bodies, such as the structure of cell membranes. So that's the second thing that the cell membrane is made of. The third thing that the cell membrane is made of is proteins. The proteins in the cell membrane do a lot of functions. First, we need to know that the proteins can be either peripheral or they can be integral. To remember the difference between those, I try to remember that peripheral sounds a little bit like peripheral vision. And if something is in your peripheral vision, it's at the edges of your vision, right? It's at the very outer edges. And so that's what peripheral proteins are like. They're at the very edges of the cell membrane. Then for integral, I try to remember integrated. They're supposed to be embedded in the cell membrane. They're going to be embedded in the cell membrane. Okay. Now let's take a look at the functions of the different proteins. So we need to know that the proteins, they're going to help with transport. They can act as enzymes. They can help with signal transduction, cell-to-cell recognition, intercellular joining, and also attachment to the cytoskeleton and extracellular matrix. So let's take a look at all 6 of those functions. We need to know that some of the proteins in the cell membrane will help with transport. They allow things to move into and out of the cell. Some of the proteins you can see can act like an open hallway, and things will just move easily in and out. And then some of the proteins will pick up molecules on one side and then flip over and drop them off on the other side. Then some of the proteins embedded in the cell membrane can act like enzymes, and they can help convert one substance to another. This is actually what you would see inside of the small intestines of humans and animals. Is inside the small intestine, there are the structures called villi, and the villi are made up of cells. And if we were to zoom in on the cell membranes that make up the villi, we would see these. We would see enzymes built into the cell membranes of those cells that are going to be breaking down things like carbohydrates and proteins. So these, this is kind of linking a little bit to anatomy, but the enzymes that are built into those cell membranes of the cells that make up the villi, these are called brush border enzymes. So that's the second function of the proteins in the cell membrane. A third function of proteins in the cell membrane can help with signal transduction. So what that means is that some of the proteins in the cell membrane will receive chemical messages, for example, through hormones, and then they will pass that message to the inside of the cell. So if you're familiar with like G proteins and second messenger proteins, or if that sounds vaguely familiar, that's what this is talking about. Because some hormones can travel straight through the cell membrane and some can't. So the ones that can't, if they're trying to send a message to the inside of the cell to tell it to make a certain protein, to stop making a certain protein, in order to send a message to the inside of the cell, that hormone may have to bind to a protein in the cell membrane, and then the cell membrane, or I'm sorry, the protein will then start a chain reaction on the inside of the cell that will eventually do whatever the hormone was supposed to do. Now let's move to cell recognition. One thing we need to know is that on the outside of all cells, there are what are called glycolipids and glycoproteins. The glycolipids and glycoproteins will very much act like ID tags. If you think about how if you're going to work, many times you'll have an ID badge. And the ID badge has to be on the outside of your clothes, right? So that way people can see it and they know who you are, whether you belong there. And so glycolipids and glycoproteins do the exact same thing. They are like ID tags and they say what kind of cell this is, what organism it belongs to, like what kind of species, and even more specifically, the individual it belongs to. So a glycolipid is almost like a way of saying, hi, my name is Leah's epithelial cell from her skin. Or, hi, my name is an E. coli bacteria flagellar cell, or flagellar protein. That's how specific they are. And this is what our immune cells are actually looking at. The glycolipids and glycoproteins are antigens, essentially. That's a little bit connecting to anatomy again, but the antigens are the foreign substances. Well, the glycolipids and glycoproteins can be antigens. So our white blood cells can look at the glycolipids and glycoproteins, and if they recognize them as ours, then they know, okay, you're a friendly cell, I'm not going to kill you. But if they recognize it as foreign or don't recognize it, they've never seen it before, then they know, okay, time to die. You don't belong here. So, right, the glycolipids and glycoproteins are very specific to species. So, the glycolipids and glycoproteins of fungi are different than the glycolipids and glycoproteins of jaguars, which are different than the glycolipids and glycoproteins of humans, which are different than the glycolipids and glycoproteins of sharks. Every single species has different glycolipids and glycoproteins. Individuals, so my glycolipids are different from your glycolipids, which are different from Harrison Ford's glycolipids, which are different from Julia Roberts' glycolipids. Everyone's glycolipids and glycoproteins are different and unique. This is actually, on that same note, this is why when we're doing organ transplants, that what scientists and lab technicians have to do is what's called type and match, I believe it's called. And technically, what they're doing is they are looking at the glycolipids and glycoproteins of the individuals. Cross type and matching. There we go. That's the specific term, cross type and matching. Because the more similar the glycolipids and glycoproteins are between the donor and the recipient, the less likely the person's immune system is going to know, ooh, you don't look familiar. Basically, the more similar that they are, your immune system goes, well, those kind of look like my glycolipids and glycoproteins. Okay, close enough. And it won't reject the organ. But the more differences that they are, the more easily that your immune system is going to recognize it and go, oh, whoa, hold up, who is this? You don't belong here. And so then it would start to attack that foreign organ and reject the organ. And this is why they typically try to look for family members first, because it's more likely that their glycolipids and glycoproteins are going to be similar to yours. And then if they aren't, then you just kind of have to rely on random chance that someone else's match with yours. Okay. So some of the proteins in the cell membrane will act as receptors for those glycolipids and glycoproteins and can help recognize them and determine whether it's foreign or not. Now let's take a look at intracellular joining. So there are some proteins in the cell membrane that can help connect cells together. Think about how for epithelial cells, lots of times they have to form layers and sheets of cells. How do they stay all connected to one another? Through proteins in their cell membrane. The proteins in the cell membrane will help join and connect the cells together. Okay, now the last function of proteins in the cell membrane will help with cytoskeleton or help with attachment to the cytoskeleton. So the cytoskeleton, which we'll discuss in a separate time, is like a mini skeleton for the cell. And it has to be anchored to something. We wouldn't want the cytoskeleton just kind of floating around and shifting and changing too much. So parts of the cytoskeleton may be anchored to proteins in the cell membrane. Okay, so as a summary, we need to know that the cell membrane is made of three things. I'm going to give you about 30 seconds to try to recall what were the three things that the cell membrane was made of. Starting now, you have about 30 seconds to try to think about it. Okay, so let's review. The cell membrane is made up of phospholipids, cholesterol, and proteins. Hopefully you got all three of those right. But if you didn't, you need to go back and review what the three things the cell membrane was made of. Now let's move on and let's talk about how do things move through the cell membrane. So some things can move straight through the cell membrane and some can't. But how do we know? How do we know if it can move straight through or if it can't? Well, that will actually depend on whether the molecule is polar or nonpolar. So let's discuss this for a second. Let's recall that the cell membrane, the phospholipids are polar and they face the outside here like this, and that the inside, remember, is the nonpolar fatty acid tails. So the inside of the cell, the inside of the cell membrane is non-polar, or in other words, hydrophobic. Non-polar and hydrophobic. So we need to know that polar substances do not like things that are non-polar. So a polar substance is not going to be able to move straight through the cell membrane. It's like a polar substance would go, oh, this is a non-polar area? Oh, heck no, I'm not going in there. I can't stand non-polar. Give me away. So it's not going to be able to make it through this non-polar interior. So examples of polar substances that wouldn't be able to travel through the membrane would be things like water, ions like sodium, anything that's an ion, sodium, potassium, chlorine, calcium, magnesium, can't travel through the cell membrane. And then also even things like glucose. So then how do they get through the cell membrane? If they can't go straight through, they would need the help of a protein, specifically a transport protein, in order to be able to go from one side of the cell to the other. Now, the substances that can go straight through the cell membrane are the things that are nonpolar, or in other words, hydrophobic. They're like, oh, you're nonpolar too? Okay, cool. We're alike. Yeah, no problem. We're the same. So because they're both nonpolar, they have no problem going straight through. Okay, so examples of things that are nonpolar and hydrophobic, those would mostly be things like oxygen and carbon dioxide. So keep that in mind. That's going to come back in a few minutes. We need so that oxygen and carbon dioxide can go straight through the cell membrane, no protein required. Right, so those are some basic concepts we have to understand. You also do need to keep in mind this other information here. If you've noticed that I've been switching between cell membrane and plasma membrane, that's because they mean the same thing. So please don't get confused and think that they're separate things. Plasma membrane, cell membrane, phospholipid bilayer, that's all the same thing. Phospholipid bilayer should make sense because the cell membrane is two layers. That's where the bilayer comes in. Bi means two. It's two layers of phospholipids. Go figure. That's right, cell membrane, plasma membrane, same thing. So as we mentioned, the polar substances will need the help of a protein in order to move into or out of the cell. There's different types of transport proteins. There can be channel proteins, and then there's also what are called carrier proteins. So the channel proteins are just like an open hallway. Stuff can move in and out. They're still pretty specific. So there's like sodium channels in which only sodium could move through. There's aquaporins, as they mention here, in which only water can move through. And then there's probably things like glucose channels, etc. So they're still pretty specific, but things can move easily in and out. The carrier proteins, they have to pick up molecules on one side and then flip over and drop them off on the other side. They're also highly, highly specific for whatever they're moving. Right now, let's discuss how do they know what direction to move? Because molecules obviously don't have a brain. It's not like they're trying to move in a specific direction. So it really will depend on differences in concentration. It will depend on differences in concentration. So we need to know that molecules tend to move from areas of high concentration to areas of low concentration. And that's called moving down the concentration gradient. So I try to remember the term downhill because when you're moving downhill, you are going from a high point up at the top of a hill down to a low point at the bottom of the hill. So that can remember the high to low, and then the downhill part can help you remember that going from high to low is moving down the concentration gradient. And if you think about it, moving, like if you were on a bike and you were going downhill, you don't have to pedal. There is no input of energy. You don't have to add any energy to move downhill. So all types of passive transport, types of transport where there's no energy involved, are going to be moving down their gradient from high to low, down the concentration gradient. Okay. Now let's take a look at the first type of passive transport. The first type of passive transport is diffusion. So sometimes it's called simple diffusion. So you might hear either term. Simple diffusion is basically what we said, movement of a substance from high concentration to low concentration, and it's out without the help of a protein. No protein is required for simple diffusion. One thing again to keep in mind is that molecules are moving randomly. It's not like they're trying to move from high to low. You can almost think about it like blindfolded people. If you put a whole bunch of people that are blindfolded in a room and you start telling them to just move in any direction, just start walking, they're going to be walking around randomly. They're not going to have any idea where they're going. They're going to be bumping into people. They're going to be bumping into walls. It's a very similar process. Molecules are always in constant motion that's totally random. So what happens is, for example, if we drop an orange dye into a beaker that has a semi-permeable membrane, the molecules, as they instantly start moving, some of them just almost by luck or by chance just happen to slip through the cell membrane, just by accident, just because they're moving randomly. They just happen to slip through the cell membrane. Obviously, not all of them are going to instantly move from high concentration to low concentration. You can see that some of them are bouncing off of the cell membrane and are not moving to the area of low concentration. And then as more time passes, more of the molecules will move from the area of high concentration to low concentration. Okay, so you can see that as more time passed, more of the molecules just happen to move from the area of high concentration to low concentration as they just keep randomly bouncing around. And also, the molecules on this right side, they don't stop moving. They're going to keep moving randomly bouncing around. And so you can see that this one's bouncing off, and then this one actually happens to move backward. So what does that mean? Nothing. You know why? Because most of the molecules are still moving from high to low. That's what matters. You need to be paying attention to what are most of the molecules doing. Some of them, yes, will actually happen to just move backwards. Oh well, as long as the majority of the molecules are still moving from high to low, it's still considered diffusion, it's still considered passive. So pay attention to what are most of the molecules doing. And then when finally the concentrations are equal on both sides of the membrane, then what happens is, again, the molecules do not stop moving. They will continue to move, but they're going to move backward and forward at an equal rate. And so this is what's called dynamic equilibrium. So when we get to chemistry, you're going to hear that term again, dynamic equilibrium. And it means the same thing, moving back and forth at an equal rate. Yeah, molecules never stop moving. So they're just going to continue to move backward and forward. So notice that again, just to reiterate, the diffusion is not needing the help of a protein. Substances that undergo simple diffusion do not need the help of a protein. So important to know. And so we need to know that, remember, what were the substances that didn't need the help of a protein? Were they polar or nonpolar substances? You should say nonpolar. So, and if you remember, what were the substances that were nonpolar? Oxygen and carbon dioxide. So oxygen and carbon dioxide use diffusion to move across some membranes. And so the place where this is going to happen is in the alveoli of the lungs. So you need to know that gas exchange that happens at the alveoli is taking place by diffusion. It takes place with diffusion. Right now, let's skip down for a minute and take a look at facilitated diffusion. So remember, if we think back to a few minutes ago, the substances that do need the help of a protein, which were they? Were they the nonpolar or the polar substances that need a protein to be transported? Should be polar. Polar substances need the help of a protein in order to move from one side of the cell membrane to the other. So they're not going to use diffusion. They're going to use what's called facilitated diffusion. So facilitated diffusion is just diffusion with the help of a protein, and that's it. It is still considered passive because it is still moving from high concentration to low concentration. The only difference is that one is with a protein and one is not, between diffusion and facilitated diffusion. That's the only difference. All right, now let's go back up and let's take a look at osmosis. Osmosis, there's a couple of really important things. You need to know all parts of this definition. That osmosis is, guess what? A type of diffusion. It is a type of diffusion, and it's not just diffusion of anything, it's the diffusion of water. So that's the second part of the definition that's important. And then the third part is that it's not even just movement of water, it has to be movement of water across a membrane, a selectively permeable membrane. So all three parts have to be there for the definition of osmosis. Like I said, it's not even just movement of water. It has to be movement of water across a selectively permeable membrane. Okay, so now let's take a look at, well, how do we know what direction is the water going to be moving? So we need to know that water is going to move from a region of low solute concentration to a region of high solute concentration. And if you're confused thinking, what's a solute? Huh? Then what you need to know is that a solute is a substance that is being dissolved. So for example, sugar could be a solute, sodium could be a solute. I mean, any kind of ion, potassium, glucose, those are all solutes. Solutes are substances that are being dissolved. Now, if you're confused and thinking, well, wait a minute, isn't diffusion from high to low? One thing you need to know is that low solute is the same thing as a high water concentration. Think of it this way. Let me draw a picture really quickly. If I have some water, I'll try and draw water molecules as blue dots, and then I'll put the solute as black dots. So low solute concentration is the same thing as a high water concentration. And high solute concentration is the same thing as low water concentration. So if I drew another picture, this, again, water is going to be in the blue dots, and then the solute is going to be in the black dots. You can see that basically the ratio of the black dots to the blue dots, there's a large solute concentration and there's very little water in comparison. So we usually, I mean, yeah, like I said, usually So diffusion, it's still diffusion because it's still going from high water to low water. It's going from high water to low water. And actually, you know what? Let me switch this because I know it's backwards. Let me move it over here. There we go. And let me scoot that one over here. There we go. That's better. So it's still going from high water to low water. We just don't normally talk about it like that. We normally talk about it in terms of solute, but know both ways. Know it both ways that water, the osmosis is moving from low solute to high solute, or in other words, from high water concentration to low water concentration. Definitely know it both ways. Okay, so let's move down and take a look at an example. So here's a great example. We have a low concentration of sugar on the left side of that membrane, and then there's a high concentration of sugar on the right side of the membrane. And so what happens is that the water is moving from the low sugar concentration to the high sugar concentration. The way that I used to try to remember this is that water goes to where it's more crowded. Or I also like to say water goes to the party with more people. It's going to go to whatever area there's a higher concentration of solute, where there's more, a higher concentration. Okay, then let's take a look at now the different types of solutions. The three different types of solutions are hypotonic, isotonic, and hypertonic. Do not worry about plants. We don't need to worry about that for the TEAS. So we can just ignore that. Let's definitely focus on just the animal cells. So there's hypotonic, isotonic, and hypertonic. So for hypotonic, we need to know that I always like to write hypotonic with a big O to remember that in hypotonic, water will flow into the cell. That water is flowing in. And why is it flowing in? Is because that's the area of higher concentration. Remember that water is always going to move to the area where it's more crowded, where there's a party with more people. So it's going to move into the cell. And so if too much water flows into the cell, then the cell may burst. And if it bursts, that's called lysed. So lysing, you should remember, is bursting. Then for isotonic solutions, in isotonic solutions, iso means same, or in other words, equal. And so it's not that there's no movement of water, there is an equal movement of water. So water will actually be moving both into and out of the cell. And that will be because there's an equal concentration of solute inside and outside of the cell. Okay, then for hypertonic solutions, hypertonic means that there's a high solute concentration outside the cell. And so that's where the water is going to go. It's going to move outside of the cell. And so when the water moves out of the cell, then the cell will shrivel. Okay. So right, you definitely need to know about the hypotonic, hypertonic, isotonic, and what direction the water is flowing. All right, now let's take a look at active transport. So active transport is going against the concentration gradient. So in other words, going from low concentration to high concentration. So you can think about this as going like uphill, going up the gradient instead of going down the gradient. And if you think about it, if you're going uphill, you're going to have to input a lot of energy to go uphill. It takes a lot of energy. And so this is why it's considered active. Moving against the concentration gradient is active transport. So, right, active transport, in order to move substances against the concentration gradient, they will require energy, usually in the form of ATP. One of the examples of active transport is the sodium potassium pump. So the sodium potassium pump is going to pump 3 sodium out of the cell and bring 2 potassium into the cell. So here we have 3 sodium being loaded into a protein. And then the ATP, when it's being used, the phosphorus on the protein is going to break off of the ATP, and it's actually going to be attached to the protein. And that process of transferring a phosphate from one substance to another is called phosphorylation. Phosphorylation, transferring a phosphate from one substance to another, or from ATP usually, um, to another molecule. And when that happens, it's usually to make it change shape or give it energy. Okay, right. And so when the phosphate attaches to that protein, that's going to give it enough energy to flip over and dump the sodium out of the cell. And then 2 potassium are going to be loaded into the protein. And then the phosphate breaks off. And when the phosphate breaks off, that's going to allow the protein to go back to its original shape. And then the 2 potassium are dumped into the cell. And then out they go. Right. The last type of active transport that we need to know about is called bulk transport. So bulk transport is for large molecules. So everything we've talked about so far has been really small. Ions, water, even glucose is considered small. But things that are super large, like large proteins, um, amino acids, large carbohydrates, they're not going to be able to fit through a tiny microscopic protein. So they're going to have to be taken in by bulk transport. So because the molecules are so large and they require like large changes to the cell membrane, that's why it's considered active. So there's 2 different types of bulk transport. We need to know that there's exocytosis and then there's endocytosis. We need to know that for exocytosis, exo means exit. And that's going to be for when something is exiting the cell. Now, it could be waste that's exiting the cell, waste products that the cell has produced that it needs to get rid of, but not always. Lots of cells use exocytosis to secrete products that they have made. For example, let's think about a pancreatic cell, a digestive pancreatic cell. So the pancreatic cell makes digestive enzymes inside of itself. That's where the digestive enzymes come from, is from the inside of cells. So then it's like, well, wait a minute, how do they get from the inside of the cell to, inside like the small intestine? The pancreatic cell, after it makes those digestive enzymes, it will package them into vesicles. Vesicles are like, I sometimes think of them as like little suitcases, just like how a suitcase you can carry and travel and it's transportable. That's what a vesicle is. It's going to contain something on the inside and it's going to travel from one place to another. So the pancreatic cell will package up those digestive enzymes into a vesicle, and then that vesicle will travel to the cell membrane and it will open up and dump it out of the cell by exocytosis. And then those digestive enzymes are going to be released into a pancreatic duct, and then the pancreatic duct will travel to the small intestine, and there you go. So any cell, hormones, or like endocrine cells that make hormones, they release those hormones through exocytosis. There's lots of cells that will use exocytosis to release their contents, something that they have made. Okay, now let's take a look at endocytosis. For endocytosis, endocytosis is going to be where something is being brought into the cell. So there's three different types of endocytosis. There's phagocytosis, penocytosis, and receptor-mediated endocytosis. So for phagocytosis, you can try to remember that the PH in phago sounds like the F sound in food. And then, so basically phagocytosis is cell eating. Then for penocytosis, I try to remember that penocytosis sounds a little bit like a pina colada, the drink. And that's how I remember that it's cell drinking. Okay. And then lastly, receptor-mediated endocytosis. It sounds complicated, but it really actually is not. Let's actually go down and let's take a look at receptor-mediated endocytosis. So for the receptor-mediated endocytosis, we need to know that, let's use an analogy. Let's imagine that we're a teacher and we work at a school and the teachers have to use a specific staff entrance. But in order to use the staff entrance, they have to get their ID card, touch it to a specific scanner beside the door, and if the scanner recognizes their ID card, then the door will unlock and the teacher can go inside. So in a similar way, there's a specific molecule called a ligand. That's like the ID card. When the ligand touches a specific receptor on the cell membrane, so that's like the scanner, the receptor's like the scanner, if they match, then it's like the cell unlocks. and it will bring the molecule inside by endocytosis. The same way that the scanner unlocked the door and then the teacher could go inside. That's all that it means. Okay, that is it for cell membrane and cell transport. But now let's get ready for a practice quiz. Okay, let's go over some practice questions. I'd like for you to answer this particular question, and I'm going to give you about 30 seconds to answer this question. One thing that you have to do, whether you're taking the TEAS or taking the HESI, you have to be good at answering questions at a reasonable rate for time management. So make sure that as you're doing your homework or practice questions or practice tests, that you set a time limit for yourself. Maybe you should give yourself some extra time the first time that you're reviewing the material or completing the homework. But then as you're repeating the homework, maybe redoing it, maybe completing more practice questions, make sure that you're answering the questions. Usually about for the HESI, usually about 1 minute per question or less. And the TEAS is about the same. You should definitely be aiming for less than a minute per question for the TEAS. And the faster that you complete some questions, then that may give you a tiny bit more time on other questions that you struggle with. But I'm going to give, actually, you know what? I'll split the difference and I'll give you about 45 seconds to answer this question, starting now. Okay, the time is up. So let's go ahead and review the correct answer for this question. Okay, so it says, which of the following statements about osmosis is true? The correct answer is C. Remember, you have to know all parts of the definition of osmosis. That osmosis is supposed to be the movement of, or the diffusion of water across a selectively permeable membrane. Has to be all parts of that definition, remember? If you picked answer choice D, you have it confused. Remember, it's from low solute to high solute. Low solute to high solute, which is the same thing as high water. So if that answer choice had said high water concentration to low water concentration, that would have been correct. But because it says solute, it's backwards. It's low solute to high solute. B is not correct. It's not the diffusion of salt, it's the diffusion of water. And we don't really have to worry about plant cells. So A is not really a concern. Let's look at another question. I'll give you about 45 seconds to answer this next question, starting now. Okay, 45 seconds is up. So let's go ahead and take a look at the correct answers. Which of the following statements about diffusion is true? Select all that apply. This is more important for the T's. The T's will have questions that are select all that apply. So HESI students, don't worry about this, but it's just good practice anyway. A is not correct. Diffusion is not active. It is passive, which means that the last answer choice is correct. B, osmosis is an example of diffusion? Yes. Remember, the definition of osmosis includes the word diffusion in it. That is the diffusion of water. So yes, it's an example of diffusion. C, diffusion is the net movement of molecules from low to high? Nope. It should be from high to low. And remember, I mentioned this example that diffusion or gas exchange is diffusion. The diffusion of oxygen and carbon dioxide occurs at the alveoli in the lungs as part of gas exchange. You absolutely have to remember that example, that gas exchange is diffusion. Very important to remember. Okay, now give me one moment and I'll bring up some other questions. Okay, I found one last question for us to try. You're going to have 45 seconds to answer this question. This question might be a little bit trickier, but it's really important to know for both the HESI and the TEAS. So you have 45 seconds to answer this question, starting now. Okay, time's up. The correct answer is A, phagocytosis. It is extremely important, especially for the HESI, that you need to know that there are some biology questions in the biology section that will connect back to anatomy. Super important to know. And vice versa, there might even be some questions in the anatomy section that cross back over to the biology section. You might think, wait, what? This seems like biology. Why is this in anatomy? Just be prepared for that because the biology and anatomy are very closely linked together. How cells work is related to anatomy. For the T's, you just need to know this in general for biology or anatomy. So keep in mind that phagocytosis is eating, and there are lots of white blood cells that will use phagocytosis to digest bacteria or dead cells or debris. And especially macrophages. Macrophages are infamous for this. In fact, it even has phage in it, which is supposed to mean eating. Neutrophils do have some phagocytic tendencies. They do tend to sometimes eat foreign substances that they've consumed. So just keep that in mind that in general, white blood cells will use phagocytosis in order to digest bacteria that they've eaten. All right, we'll stop